Today we will learn the chapter 9. Chapter 9 title is Protein Expression, Purification and Characterization. Okay, so chapter 9 is composed of 3 subjects starting from the recombinant DNA technology and protein expression, and second subject is harvesting, and final subject is purification. Okay, so in this slide we will learn the first subject, recombinant DNA technology and protein expression. So this procedure is belong to the cell line development. Okay. So in this figure, I will show you the biopharmaceutical manufacturing process, which is starting from the cell culture and centrifugation and purification and the packaging. The first step we call it as cell culture. Okay, for the cell culture, first of all, we have to make the cell line. So this is the procedure to make the cell line. So this procedure we call it as the cell line development. Okay, first of all, we have to find our gene of interest. Okay, so if you find if you want to make the insulin, so you have to clone the insulin gene first. Okay, so this process we call it as gene of interest optimization. So after that, we have to make the vector. Okay, so we have to do several cloning. So finally, we will make the expression vector. After that, we have to do transfection and selection and growth. So this is growth is first of all we can do the growth at flasker. So we can do the flasker. After that, we can do the scale up. So finally, we can culture our cell line in the big bioreactors like this. Okay. The most important thing in cell culture is a cell line development until here. Okay. So the flasker this step is very important. So who is doing this research until here? Who is doing this research? So it's me. So if you are interested in this cell line development, please contact to me, okay? In this slide, I will explain how to construct a vector for the production of a therapeutic antibody, okay? First of all, I will explain the vector structure. So vector is composed of the 3 important factors. The first one is the origin of replication, and second one is the antibiotics resistance gene. The final one is multiple cloning site. So let me explain the origin of replication first. So this site is the origin of replication. So we call it as ORI. So this is the site where DNA replication begins. So usually this is the origin of replication. First of all, initiate transcription factor bind to here, and after that, helicase is unwinded double-stranded DNA. So after that, they will make the 2 independent. They will replicate the plasmid. So one plasmid will become the 2 plasmid. 2 identical plasmid is like this. Okay. Second one is the antibiotics resistance gene, like this. So this antibiotic resistance gene allows bacteria containing a vector to grow on a selective media. For example, if they have the ampicillin resistance gene, it means that this bacteria, the bacteria containing a vector, can grow on a media containing the ampicillin. Okay, so if they have the tetramycin, for example, tetracycline resistance gene in the vector, it means that the bacteria containing a vector to grow on a selective media, for example, containing tetracycline. Okay. So final important factor is that multiple cloning site. So this is a multiple cloning site. If you see here multiple cloning site, contains multiple enzyme cutting sites. For example, they are composed of the ECOAL1, HIND3, SAL1, JO1, KPN1. Finally, they are composed of the BGL2 enzyme cutting site. Okay, so this is the important criteria in the vector structure. Once again, origin of replication. And second one is the antibiotics resistance gene. And final one is the multiple cloning sites. So we can do cloning in our gene of interest in these multiple cloning sites. Okay, in addition to three important factors, and vector should have another important structure. The first of all, they should have the inducible promoter. Okay, so inducible promoter is composed of the I region and promote region and the operate region. Okay, so this is recorded as inducible promoter. So I region can be transcribed, so it will make the mRNA. So mRNA can be translated, so it will make the repressor. So usually the repressor protein can bind to the operate region. So when repressor can bind to the operate region, so there will be no transcription. Okay, so let me show you one example. So this is a symptom of diabetes. So usually the diabetes patient have a difficulty to manage their blood sugar level. That's why they have to inject insulin by themselves every week. Okay, so how to make the insulin? Okay, so this is vector structure for the production of the insulin. So they use the inducible promoter. Okay, first of all, inducible promoter, we need the inducer. So this is the structure of the inducer. The full name of the inducer is isopropyl. So this is isopropyl thiogalactose. So that's why we call it as IPTG. Okay. So in the absence of the inducer, in the absence of the IPTG, so repressor is bind to the operate region. So it will prevent the transcription. So it will prevent transcription. However, in the presence of the inducer, so inducer can bind to the repressor. After that this repressor cannot bind to the operate region. So it means that several transcription factors can bind to the promoter region. So after that, so insulin gene can be transcribed. So it will allowing transcription to proceed. Okay, so IPTG bind to the repressor and means that the repressor cannot bind to the operate region. So it means that several transcription factors can bind to the promote region. After that, insulin gene can be transcribed and finally it will translate. Okay, so for the production of insulin, vector also need special characteristics. First one is prokaryotic ribosome binding site. So we call it as a Shine-Dalgarno sequence. So this is the vector sequence which is composed of the promote region, and they have the protein coding region. Okay, starting from the start codon, finally they have stop codon is here. So in front of start codon, so vector need Shine-Dalgarno sequence. Okay, so if this DNA is transcripted, so this is the mRNA, so this is start codon AUG. Okay, so Shine-Dalgarno sequence is the AGG So this is the Shine-Dalgarno sequence. Usually the ribosome can bind to the here. After that, translation is happen from the start codon. Okay, and also vector also need transcriptional terminator at the end. Okay, after the stop codon, So they need a vector need transcriptional terminator, which is composed of the self-complementary sequence, FDAT, and poly A tails. Okay, so this transcriptional terminator mediates transcriptional termination by synthesizing hairpin structure is like this. After that, this hairpin structure can be dissociated from the DNA after that transcription termination happens. Okay. In this slide, I will explain the cloning. So what is cloning? So cloning is process of constructing new DNA molecules. So once again, cloning is process of constructing new DNA molecules like this. Okay, so this is the cloning step, starting from the prepare foreign DNA and the cloning vector. So cloning vector has the MCS, multiple cloning site. Okay, after that, use restriction enzyme to generate DNA fragment. After that, each restriction enzyme make the generate the DNA fragment, for example, foreign DNA fragment and digested vectors like this. After that, anneals foreign DNA fragment to digested vector. After that, ligate. Okay, once again, so anneals foreign DNA fragment to the digested vector. After that, ligated. So finally, we will have the assembled DNA is like this. So this is the cloning procedure. Okay, starting from the prepare foreign DNA and cloning vector has the multiple cloning site. After that, use the restriction enzyme to generate DNA fragments. After that, anneals foreign DNA fragment into the digested vector. After that, ligated. So this is the cloning procedure. Okay, so in this slide, we will learn the basic principle of the restriction enzyme and also basic principle of the ligase. Okay, so what is restriction enzyme? What is their function? Their function is they can recognize specific DNA sequence, and after that they can catalyze their cleavage. Okay, this is a table 7.7 to show that several restriction enzyme and also their recognition site. Okay, for example, EcoR1, this enzyme can recognize the GAATTC. After that, they will induce cleavage between the guanine and adenine. Okay, and also all of the restriction enzyme is belong to the hydrolase. Okay, they usually use the water to break the chemical bond. And for example, EcoR1 recognize the GAATTC, and after that, they can do cleavage between the G and A. At that time, they use the water to break the chemical bond. And also, what is ligase? So previously we learned that this is the ligase. Okay, so at the final step, so anneals foreign DNA fragment to the digested vector and ligate. Okay, at this step, ligase plays important role at the final step. Okay, so usually the ligase use the ATP. Okay, after that, they will make the phosphodiester bond linkage between the nucleotide and nucleotide. Okay, once again, the ligase use the ATP energy. After that, make the phosphodiester bond linkage between the nucleotide and nucleotide is like this. In this slide, I will explain the second step in the biopharmaceutical process. We call it as the harvesting. harvesting is starting from the cell wall disruption. For example, to make the insulin, we always use the gram-positive bacteria. So in this case, for example, gram-positive bacteria has the several peptidoglycan layer exist outside the cytoplasmic membrane. So this is the cytoplasmic membrane. So outside the cytoplasmic membrane, there is very thick peptidoglycan layer is existed. Is like this. So we have to. So this is very strong. So we have to apply mechanical disruption. First of all, we always use sonication. So sonication means that sonication is the act of the applying the sound energy. After that, it agitates particle in a sample. So finally, cell wall can be disrupted. Okay, second one is we always use glass bead. Okay, so this is the glass bead and this is the bacteria sample. So glass beads collide with the cells and cracking cells to release intercellular component. So, okay, once again, so the first step in the harvesting is that cell wall disruption. For example, gram-positive bacteria, we always apply mechanical disruption. For example, sonication and glass beads. Okay, so secondly, the gram-negative bacteria. For example, this is the gram-negative bacteria, which is composed of the out membrane and also cell membrane. Between the out membrane and the cell membrane, so there is a peptidoglycan layer is existed. So peptidoglycan layer is composed of, peptidoglycan layer is composed of the NAM, NAG, NAM, NAG, NAM, NAG structure. So this is the NAM, NAG structure is like this. Okay, so NAM is also based on the glucose, and the NAG is also based on glucose. So between the NAM and NAG, there is the beta 1, 4. This is a 1, and this is first carbon, and this is fourth carbon. So beta 1, 4 glycosidic bond is existed between the NAM and NAG. So usually the lysozyme, so this enzyme is disrupted, so it can disrupt the beta 1 and 4 glycosidic bond in the NAM NAG unit. So in the case of the gram-negative bacteria, we always apply the lysozyme. So lysozyme can hydrolysis of the beta 1 and 4 glycosidic bond in NAM NAG unit. Because we can use the lysozyme because the especially the gram-negative bacteria, we only have very thin layer of the peptidoglycan, the layer. However, for example, gram-positive bacteria, they have a very thick layer of the peptidoglycan layer, so we cannot use the enzyme method. We have to use the mechanical method. Okay. So in this slide, I will explain the second step in the harvesting. So we call it as centrifugation. Okay, so after cells were fractionated, okay, that we apply the low speed centrifugation. So centrifuge at the low speed, after that we can remove the unbroken cells. So unbroken cells can be go to the pellet, and also all cell content and membrane and organelle will belong to the supernatant. Okay, so after that, progressive increase in the centrifugal forces, okay, we can remove the large membrane and the nuclei, and also we can use a supernatant, and after that we can do the progressive increase in the centrifugal forces. So eventually, at highest speed, So protein complex can be separated. So this is the centrifugation. So in this slide, I will explain the mathematics in centrifugation, especially focus on how to calculate relative centrifugal force, RCF. So RCF means that centrifugal force divided by force due to the gravity. So finally, the RCF is the omega 2 factorial multiply R. R means that rotor diameter, that this is G, divided by G. Okay, so omega means that angular velocity. So angular velocity means that radians per second. So it means that 2 multiply phi. Phi is 3.14, something like that, and multiply is RPM. So RPM is the number of turns in 1 minute. So that's why we have to divide it by 60. So this is the omega, the angular velocity. Omega is 2 pi multiply RPM divided by 6. At that, okay, so let me show you one example. So what is the average relative centrifugal force in a 10 centimeter rotor spun at 10,000 RPM? Okay, so this is the RCF is that 2 pi multiplies the RPM is the 10,000 divided by 6, and at that 2 factorial and multiply the rotor diameter is the 10 centimeter, so it will be 0.1 meter and divided by 9.8. So it will be the centrifugal force, relative centrifugal force will be 11190. Okay. Okay, so in this slide, I will explain the third subject, purification. Before we further study the purification, let me start from the ionic strength. So ionic strength, we call it as I. So usually the ionic compounds, when dissolved in the solution, so usually the ionic compounds dissociate into ions. For example, cation and anions. Cation, they have plus charge. So anion, they have minus charge. So ionic strength is the measure of the concentration of the ion in the solution. So this is the how to measure, how to calculate ionic strength. Okay, so M means that this M plus means that concentration of the cations, and the Z plus means that charges of the cations. So usually the Z 2 factorial. Okay, and also plus M minus means that concentration of the anion, and the Z minus means that charges of the minus ions at that 2 factorial. At that we just divide by 2. So this is the ionic strength. Okay, so this is one example. So what is the ionic strength of a 0.75 mole solution of the MgCl2? Okay, so let me calculate first. Okay, for example, MgCl2 is dissociated with the Mg2 plus and Cl minus. So what will be the concentration of the Mg2 plus? It will be the 0.75 mole. How about the charges of the Mg2 plus? It will be plus 2. How about Cl minus? What is the concentration of Cl minus? Because they have the Cl2, it means that their if the MgCl2 is dissolved in the solution, so it will make the 2Cl. So it means that their concentration is 2 multiply 0.75 mole. So what is the charges of Cl minus? So it will be -1. Okay, so the ionic strength will be the first concentration will be 0.75 multiply 2 factorial plus Cl minus concentration will be the 0.75 multiply 2 after multiply -1 2 factorial after divided by 2. So the ionic strength will be if after calculation you will find that the ionic strength of the 0.75 mole solution of MgCl2 will be the 2.25. It's like this. Okay, so in this slide I will explain the first step in the purification. Okay, so the first step in purification, we call it as a salting in and salting out. Okay, let me explain the salting in first. Okay, so previously we learned that hydrophobic effect. Okay, so in the presence of the water, so usually the hydrophobic amino acids, for example, hydrophobic amino acids clumps together. So this is, we call it as the hydrophobic effect. So usually the protein in the solution, so hydrophobic effect happens, so they clumps together like this. Okay, so when salt at the low concentration, so within this concentration, salt within this concentration, when is dissolved in the solution, so it will have the low ionic strength. Okay, so salt ion at the low ionic strength can neutralize charges on protein space. So protein space has the positive or negative charges. So salt ion at the low ionic strength can neutralize charges on protein space is like this. Okay, so if the salt concentration increased, so in this case is the, for example, salt ion is the neutralized nearly all of the charges on the protein space. So when this happens like this, okay, so it means that when the protein is clumps together, so there are lots of the protein electrostatic interaction. However, when this happens, okay, so it means that it will decrease the protein electrostatic interactions. So it means that when this case is protein is clumps together, but it means that protein solubility is very low. But when this case happens, so it will increase protein solubility. It's like this. So this is the salting in procedure. Okay, so in this slide, I will explain the salting out procedure. So when salt at high concentration, it means that the concentration is above the certain point. So when salt at high concentration is dissolved in solution, it means that some ion can neutralize charges on protein. However, many ions do not bind to the proteins, so it will make the excess ions like this. So usually the excess ions compete with the proteins for the solvent. So it means that resulting in the decrease of protein solubility. So finally, the decrease in protein solubility allows the protein to aggregate, and finally the precipitate. So this is the salting out procedure. So salting in, salting out procedure is the first step in the purification. So in this slide, I will explain the cosmotropes. Okay, what is cosmotropes? So this is cosmotropes ion. Okay, so usually the cosmotropes ion exhibit strong interactions with the water molecules. Okay, so this is water molecules like this. Okay, this is water molecules. So usually the cosmotropes ion exhibit strong interaction with the water molecules. So like this. So usually the cosmotropes ion is attract all of the surrounding water molecules is like this. So it means that it will make the hydrophobic effect here. So it means that proteins can be aggregated like this. So proteins can be aggregated like this because it will increase the hydrophobic effect within the solution. So it means that cosmotropes ion causes protein to precipitate in protein's native structure. So if we increase the cosmotropes ion concentration, it means that this protein can be precipitate in protein's native structure. So this is the cosmotropes. Once again, cosmotropes exhibit strong interaction with water molecules, resulting in the increase of the hydrophobic effect within solution. It means that protein can be precipitated in protein's native structure is like this. Okay, so in this slide, I will explain the chaeotropes. So what is chaeotropes? Okay, so this is the chaeotropes ion. Okay, this is a chaeotropes ion. Okay, so chaeotropes ion disrupt the hydrogen bondings between the water molecules. So this is water molecules. So this chaeotropes ion disrupt the hydrogen bonding between the water molecules. It means that resulting in the decrease of the hydrophobic effect within the solution. Okay, so it means that decrease in the hydrophobic effect around the hydrophobic amino acids. So it means that cannot make the hydration shell around the proteins. So when the hydrophobic effect is large or when the hydrophobic interaction is high, so protein can combine together like this. Okay, so this is water. So this is we call it as the hydration shell. Okay, when the decrease in hydrophobic effect around the hydrophobic amino acids, so cannot make the hydration shell around the protein. It means that it will decrease the making of the hydration shell. It means that the protein, it means that so there will be no hydration shell around the proteins. It means that protein can be denatured like this. Okay, so if we did sorting out the experiment, so chaeotropes cause protein to precipitate in denatured structures like this. Okay, so this is we call it as the chaeotropes. So in this slide, I will explain the stabilizing property of cosmotropes and chaeotropes. The previous slide, we learned that cosmotropes ion cause proteins to precipitate in native structure, so their stabilizing property is very high. However, chaeotropes ion is cause protein to precipitate in denatured structure. So it means that their stabilizing property is very low. For example, this is a cation plus charge, and this is anion which has a minus charge. For example, this one is cosmotropes ion, and the final one is the chaeotropes ion. Okay, in the anion and sulfate is the cosmotropes ion, and the thiocyanate is the chaeotropes ion is like this. Okay, the very famous cosmotropes ion is belong to the ammonium. Okay, ammonium NH4 plus, and also sulfate SO4 2 minus. So this is the very famous cosmotropes ion, and also very famous cation is the guanidinium, and also thiocyanate is like this. Okay, So once again, the stabilizing property of cosmotropes and chaotropes. Cosmotropes ion, they have their stabilizing property is very high, and however, chaotropes ion is the cause protein to precipitate in denatured structure. It means that their stabilizing property is very low. It's like this. Okay. This slide I will explain the reason to use ammonium sulfate NH4 to SO4 in protein purification. So in the company, for example, Celtrion and Samsung Biologics, during the purification step, we always use ammonium sulfate. Why do we use ammonium sulfate for protein purification? Okay, I will show you the reason. Okay, so this is the figure 9.11. The solubility of the hemoglobin in different electrolytes, in different salts. Okay, for example, the y-axis is the log solubility, and x-axis is the ionic strength. So ionic strength means that salt concentration. Okay, so the difference between this graph and this graph is that, so in this graph, the y-axis is the solubility. So in this graph is the y-axis is the log solubility. Okay, so this graph is also show that salting in, salting out result. Okay, so this is the we use the salt as the NH2SO4, and this graph we use the ammonium sulfate, and this graph we use the KCl, and this graph we use the NaCl. So after that we checked the solubility of the hemoglobin. Okay, so we checked solubility of the hemoglobin. Okay, so if you see this graph, so ammonium sulfate, so this is ammonium sulfate. So ammonium sulfate is very soluble in the water, and also ammonium sulfate can reach at high concentration. For example, ammonium sulfate can reach higher concentration. So it means that resulting in complete precipitation of proteins. So when this ionic strength, the hemoglobin's solubility is nearly zero. It means that resulting in complete precipitation of the hemoglobin. That's why we always used ammonium sulfate in the protein purification. So the second reason to use ammonium sulfate in protein purification is that ammonium sulfate is composed of the ammonium ion and sulfate ion. So ammonium ion and sulfate ion belong to the cosmotropes ion. It means that cosmotropes ion exhibit strong interaction with the water molecules. So this is the cosmotropes ion. So exhibit strong interaction with water molecules. So it means that it will increase the hydrophobic effect within the solution. So it will increase the hydrophobic effect within the solution. So it means that both ion can cause protein. So this protein can precipitate in native structure. That's why we use the ammonium sulfate in protein purification. So because we can precipitate, we can cause proteins to precipitate in native structure. That's why all of company use the ammonium sulfate in protein purification. So in previous slide, we learned about salting in and salting out procedure. Through this procedure, we can only precipitate proteins from the solution. Okay. After that, we just collect all of the protein sample again. After that, we redissolve that protein. So after that, we always do the chromatographies. So chromatography is the very important step in the purification. So if you go to the Celtrion and or Samsung Biologics, all of you guys will go to the purification group. So you will do the chromatography. Every day you will do the chromatography. Okay. So let me see the in this slide I will explain the ion exchange chromatography first. Okay, so ion exchange chromatography can separate proteins according to the overall charges, whether they have plus charge or whether they have minus charges. Two different kinds of the ion exchange chromatography is available. First one is anion exchange chromatography, and second one is cation exchange chromatography. So, for example, the anion exchange chromatography, the molecule of interest is anion, so negatively charged. How about cation exchange chromatography? The molecule of interest is cation, the positively charged. Okay, so this picture to show that one example, ion exchange chromatography, especially the anion exchange chromatography. Our molecule of interest is the anions, minus charges, so negatively charges. So this is a mix of the amino acid. So okay, the column is positively charged. So this the ion exchange column is positively charged. So mix of amino acid can get poured into the column, but our molecule of interest is anion, negatively charged. That's why we call it as anion exchange chromatographies. Okay, so in this case, only minus charged amino acid can bind to the resin, positively charged resin. Okay, how about the positively charged amino acid? This amino acid can pass the column. Okay, so finally, we only can collect the negatively charged amino acids. Okay, once again, our molecule of interest is the anion, negatively charged. That's why we call it as this chromatography is the anion exchange chromatography. So in this slide, I especially focus on the anion exchange chromatography. Okay, so anion exchange chromatography has a cationic resin. They have a positively charged resin is like this. Okay, so figure 9.16 to show that the common functional groups found in the ion exchange resin. Okay, the very famous the cationic resin is the QAE or DEAE. Okay, the first one is QAE. The full name is the quaternary amino ethyl. Quaternary, first, second, tertiary, quaternary amino ethyl. So they have a positively charged is like this. Second one is the DEAE. So DEAE has the full name is diethyl amino ethyl. The full name is the diethyl amino ethyl DEAE. So usually, and also they have positively charges like this. Okay, so QAE is strongly bind to the negatively charged protein. Okay, because they have positively charges, and also DEAE has the weakly bind to the negatively charged protein because they also have positive charges. They are positive charges, so but they can weakly bind to the negatively charged protein. So depending on the proteins, so sometimes we use QAE and sometimes we use the DEAE, but most of the cases we use the QAE. Okay, so in this slide, I will especially focus on cation exchange chromatography. Okay, so this is cation exchange chromatography. So they have the resin. Okay, this is resin, but they have the negatively charged resin. So we call it as the anionic minus charged resin. Okay, so there are two famous negatively charged resin is available. The first one we call it as SP, and second one we call it as CM. Okay, so this is the first one. The full name of the resin SP is sulfopropyl. Sulfopropyl. Okay, so we call it as SP. So they have the two minus charges like this. Okay. Second one is we call it as the carboxyl methyl, and the name is we call it CM. Okay, full name is carboxyl methyl. So they have a carboxyl methyl. Okay, so they have the one minus charge is like this. Okay, the sulfopropyl SP can strongly bind to the positively charged proteins because they have the two negative charges. Okay, and also CM carboxyl methyl can weakly bind to positively charged protein. So they have only one minus charges. Okay, so CM can weakly bind to positively charged protein. So most of cases we use the SP resin, but sometimes we use the CM. Okay, but most of cases we use a strong anionic resin. The full name is the sulfopropyl. So like this. Okay. Okay, so in this slide, I will explain the size exclusion chromatography. So another name we call it as gel filtration chromatography. Okay, so this is the size exclusion chromatography. So this chromatography can separate proteins according to their size. Is like this. Okay, and also the chromatography, this size exclusion chromatography is composed of the particle, because this is particle which has pore inside. They have pore inside. Okay, so for example, large proteins cannot enter the particle pores. So usually the large protein can be excluded, right, like this. So it means that large proteins elute at early stages. So large protein can be eluted at early stages. And also large protein shows that faster rate of progression. Okay, so large protein shows the fast rate of progression. Okay. However, small protein can enter particle pores. So it means that they can enter particle pore, and they can enter particle pore, they can enter particle pore, so they can enter particle pore. So it means that elute at late stages. So they will elute at late stages. So that's why the small protein exhibit slow rate of progression. Okay, so this is the principle of size exclusion chromatography. So okay, this chromatography can separate proteins according to their protein size. So large protein elute at early stage, and small proteins elute at late stages. So in this slide, I will explain the estimation of molecular mass using size exclusion chromatographies. Okay, so this is the graph to show that y-axis is represent elution volume, and it's also the x-axis is represent the logarithm of the molecular mass of proteins. So logarithm of the molecular mass. Okay, so usually the large protein, they exhibit fast rate of progression. Okay, so large proteins can be detected when total eluted volume is very small. Okay, it's because they exhibit fast rate of progression. So usually the large size proteins can be detected when total eluted volume is very small. Okay, so when total eluted volume is very small, we can easily detect large protein. Okay, after that, however, small proteins which exhibit slow rate of progression, so can be detected when total eluted volume is very large. Okay, so small proteins can be detected when the total eluted volume is very large. So when the total eluted volume is very large, so in this case we can detect the small size proteins. And people found that elution volume, total elution volume, and the logarithm of the molecular mass, elution volume and the logarithm of molecular mass shows that linear relationship. For example, if the elution volume is around here, their molecular weight is around here, and also the elution volume is around here, their molecular weight is here, and elution volume is around here, and we found that their molecular weight is around here. Okay, so based on this linear relationship, negatively linear relationship, so we can estimate molecular mass by using the total eluted volume. Okay, so when the total eluted volume is, for example, around 2 ml. Okay, so when the total eluted volume is around 2 ml, so based on and that we can estimate molecular mass like this. So maybe at this location, the protein size will be like this. Okay, so this is once again the estimation of the molecular mass using size exclusion chromatography. So because finally we can estimate molecular mass, for example, around here is 3 ml, total is 3 ml, so maybe the 3 ml will be like this. Okay, so we can estimate the proteins, the size here. Okay, so this is the estimation of molecular mass using size exclusion chromatographies. In this slide, I will explain the purification of small size proteins using size exclusion chromatographies. Okay, so this is graph. So this is the x-axis is a logarithm of the molecular mass, and the y-axis is the elution volume. Okay, so maybe the small size proteins will be located within these ranges. Okay, so it means that they are not the small size protein is not within the linear curve. Okay, so it means that even after a certain amount of running buffer has run out in the column, but you cannot estimate whether you can get the small size proteins. So because they are not within the linear curve, so in these cases, we always add polyethylene glycol in the running buffer. So this is the ethylene glycol structure. So polyethylene glycol, PEG, is so this unit is the multiplied. So N is like this. So this ethylene glycol is multiplied. So this is recorded as polyethylene glycol. So for example, this is proteins. So protein has an N-terminal region. So usually the polyethylene glycol is bind to the protein. It's like this. Okay, so this is the PEG-related protein. So usually the PEG can attach it to the protein, especially the N-terminal region. So finally they will make the PEG-related proteins. Okay, so it means that the size of the PEG-related proteins will be within the linear curve. Okay, it means that after certain volume of the learning buffer has running out in the column, so maybe we can get the protein that we want to purify. So this is the we always used when we purify small size proteins. So once again, so their size is not within the linear curve. So we always add a PEG to the learning buffer. So usually the PEG is attached to protein, especially the N-terminal region. So it means that the size of the PEG-related proteins will be within the linear curve. So it means that after certain amount of the learning buffer has run out in the column, so it means that we can get the proteins that we want to purify. Okay. In this slide, I will explain the new chromatography. So we call it as affinity chromatographies. So affinity chromatographies use non-covalent interactions between immobilized ligand and the proteins. So this is the one example of the immobilized ligands, which are composed of resin and protein A and G. Okay, so this is the immobilized ligand. So usually the antibody can bind to the AG region, protein AG, but their interaction is not covalent interaction. So most of their interaction occurs by hydrogen bonding. Okay, so affinity chromatography use once again non-covalent interactions between immobilized ligand and the proteins, between the immobilized ligand and the proteins. Okay, so after that, proteins that bind to the ligands will remain in a column. So only proteins that bind to the ligands will remain in the column is like this. Okay, so after that, we have to collect our target proteins. So in this case, we use the elution buffer. So we apply the elution buffer here. So elution buffer, we can change some of the pH. Okay, so elution buffer at specific pH can disrupt the interactions between the immobilized ligand and the proteins. So finally, we can collect the target protein that we want. Okay, so this is the principle of the affinity chromatographies. Okay, so in this slide, I will explain the protein A affinity chromatography or protein G affinity chromatography. Okay, for example, protein A affinity chromatography, especially the immobilized ligand, is composed of resin and protein A. Okay, how about protein G affinity chromatography? So protein G affinity chromatography, for example, immobilized ligand is composed of the resin and protein G. Okay, so usually the protein A or protein G is exhibit affinity for Fc region of the antibody. So this is the antibody structure, which is composed of the heavy chain and light chain. Okay, so we call it as this region as a Fab region, and also we call it as this region as the Fc region. So usually the protein A or protein G can bind to the Fc region of the antibody, is like this. Okay, so nowadays all of the Celltrion or Samsung Biologics, and they make the cell line which can make the antibody. So usually the protein A affinity chromatography or protein G affinity chromatography can separate antibody subclasses from the cell culture supernatant. So because the cell is the secreted antibody, so usually by using the protein A affinity chromatography or protein G affinity chromatography, so we can separate the antibody subclasses from cell culture supernatant. Okay, so in this slide, I will explain another affinity chromatography, especially the nickel affinity chromatography. So in the nickel affinity chromatographies, so immobilized ligand is composed of resin and the nickel, especially the nickel 2+. Okay, so this nickel 2+ ion is exhibit strong affinity for polyhistidine. So this is histidine, 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 histidine. For example, this is the 6-histidine is composed of the polyhistidine tag. So usually the nickel ion have a strong affinity for neighboring histidine, especially this is nitrogen. So between nitrogen and the nickel, so they have the non-covalent bonding is existed. Okay, so in usually whenever we purify the recombinant proteins, we use this vector which has the histidine tag. So this is the DNA encoding proteins. So we cloned DNA encoding proteins into the vector which has the histidine tag. So finally we will make the vector, the DNA encoding proteins plus histidine tags. So it means that the recombinant proteins with polyhistidine tag can be synthesized. So by using the nickel affinity chromatography, so we can separate any recombinant proteins containing the polyhistidine tag like this. Okay, so in the company, so for example, the Samsung Biologics and the Samsung Biologics and the Celtrion, they usually make lots of the vector which contains histidine. So nowadays they separate tons of the recombinant proteins containing polyhistidine tag. Okay. Today we learn the chapter 9. Chapter 9 composed of 3 subjects starting from the recombinant DNA technology and protein expression. This step is the crucial role in doing the cell culture. And also second subject we learn the harvesting. Harvesting is also the second step in the biopharmaceutical manufacturing process, in which we covered cell wall disruption and centrifugation. And third step is the purification. Okay, so we cover sorting in and sorting out. We also cover several chromatography technology. Okay, the final step in the biopharmaceutical manufacturing process is packaging. But today's lecture we will not cover this one. Okay, so today we covered cell culture and harvesting, purification. Okay, so today we will finish the class.